Jnanam paramam dhyeyam. Knowledge is supreme.
Hi, let us recap on what we looked in the last lecture. We looked at the different steps which are involved in catalytic reaction. We particularly observed there are 7 steps that are actually present, where step number 1 is the species from the bulk fluid diffuses and reaches the exterior surface of the pellet due to external diffusion, and then the species through the pores via the internal diffusion reaches the surface of the catalyst, which is actually step number 2, and the species which reaches the surface of the catalyst actually gets adsorbed onto the active sites, and that is step number 3, and the adsorbed species undergoes reaction, which is step number 4, and following which the products which are now sitting on the active sites get desorbed near the catalyst surface, and that is step number 5, and following which the product species diffuses back through the pores via the internal diffusion to the exterior surface of the pellet, and the product actually from the exterior surface diffuses via the external diffusion to the bulk fluid. So, these are the 7 steps which are involved in catalytic reaction, and we attempted to find out what is the rate of reaction for the first 2, what is the rate for first 2 steps, that is the external diffusion and the internal diffusion. Now, we will start with the third step, which is the adsorption. So, we look at step 3, which is adsorption. We want to find the rate at which the species are actually getting adsorbed from the gas phase into the solid surface, and we drew a cartoon in last lecture. We said that if this is the surface of the catalyst, and there are these sites where the species can adsorb, and if let us say that the empty hats are the vacant sites, and let us say species A, which is actually gas species, which comes and adsorbs onto let us say one of the empty sites, and let us represent it by a filled hat. So, species A is now sitting on the site which is basically filled, which means that that site is actually occupied. And so, in order to quantify this process, we define a couple of terms. We define what is called as Ct, which is the total molar concentration of the active sites. We also defined what is Cv, which is the concentration of the vacant sites. Suppose there are 2 species, 2 gas species which are present, then it could be that species B, which may also be participating in the reaction, they may come and sit in another site, they may come and occupy another site. So, let us now define concentration of species A, which is sitting on the catalyst active site, that maybe is CAS, where A plus S reversibly sits on the catalyst site. So, if CAS is the concentration of the species which is actually sitting in the catalyst site or which is adsorbed onto the vacant catalyst site, and then CBS let us say is the corresponding concentration of the species that is actually adsorbed onto a catalyst site. So, typically the adsorption is actually characterized by what is called as an isotherm. So, what we are going to spend in most of today's lecture is actually going to characterize the isotherm. So, the definition of isotherm is that it essentially refers to the amount of gas adsorbed on solid surface at different pressures and at fixed temperature. So, how do we find what is this isotherm? How do we find what is the relationship between the amount of gas adsorbed and the rate at which the species is actually getting adsorbed? So, let us look at what really happens at the molecular level. So, the gas species which is now coming and present close to the active site of the catalyst or the solid surface in the pores. So, the number of molecules which is actually present, number of gas molecules that is present, they will collide with the species at the surface which is present, the catalyst species that is present. So, clearly the adsorption that is happening because of collision is a strong function of the number of molecules of the gas species which is present close to the surface. So, clearly the rate at which the adsorption is going to happen is a function of or is proportional to the partial pressure of the gas species that is present near the solid surface. So, let us try to quantify this. So, let us take a specific example of carbon monoxide. So, there are two possible cases where you may have CO which is actually reacting with, let us say, nickel catalyst. So, let us say these are the vacant sites in the nickel catalyst, and that will reversibly adsorb onto the surface where the whole molecule itself will adsorb onto the active sites of the catalyst, or it will form a bond with the nickel catalyst which is present. There may be another possibility where CO, let us say if it is in presence of iron catalyst, then the molecule might dissociate and the species would actually go and bind to different sites. So, for instance, Fe, you may have carbon atom which is sitting here and the oxygen is sitting in another site. So, there are two different ways by which the adsorption can happen. One could be because the whole molecule by itself could actually go and adsorb onto the active site, or the molecule can dissociate itself and the individual species can go and adsorb onto the active sites. So, clearly there are two possible ways by which adsorption can happen. One is called the molecular or non-dissociated adsorption, which is essentially the first case, and then one can also have the second case where you have dissociated adsorption, which is essentially the second case. So, we will find out the rate at which the adsorption can happen for both these cases. So, let us take the first one. So, let us say non-dissociated adsorption. So, let us now say that S represents a vacant site. So, CO plus S will give COS, which is actually adsorbed onto the vacant site. So, now the rate of attachment of the species onto the active site clearly is a function of the partial pressure of the gas species which is actually adsorbing on site. So, that should be equal to K. Let us say that the attachment is 80 multiplied by the partial pressure of CO multiplied by the concentration of the vacant sites. Now, the rate of detachment will be K minus A into concentration of the species which is actually present, concentration of the species which is actually bound to the active sites. So, therefore, the rate of adsorption is essentially given by Ka PCO into Cv minus K minus A into COS. So, this we can rewrite as Ka into PCO into CV minus CCOS divided by an equilibrium adsorption equilibrium constant called Ka, which is essentially defined as the ratio of Ka by K minus A. So, this is the adsorption equilibrium constant, and now we can actually use a conservation rule to actually understand how to define the rate of adsorption. Now, if you stare at the expression for the rate of adsorption, you will see that the quantity which we do not know is essentially this vacant site concentration. So, we will try to eliminate this in order to get the rate in terms of the measurable quantities. So, now we can use the conservation rule. The conservation rule essentially is that the total concentration of the sites, active sites, should be equal to the concentration of the vacant site CV plus the concentration of the species adsorbed onto the sites. So, therefore, CV is essentially equal to Ct minus CCOS. So, now if we plug this into the rate expression, we will see that our rate of adsorption is equal to some Ka into PCO into Ct minus COS minus CCOS divided by Ka. So, now if at equilibrium the R adsorbed is equal to 0, which means that the attachment and the detachment rates are equal. So, from here clearly P times CO into Ct minus P times CO into CCOS minus CCOS divided by Ka, that is equal to 0. So, from here we can deduce that CCOS, which is the concentration of the adsorbed CO onto the active sites, that should clearly be equal to PCO into Ct divided by 1 plus Ka PCO into Ka here. So, it is just a little algebra. So, once you do the algebra, you will find that the concentration of the species that is adsorbed onto the active sites is essentially given by this expression here. So, now let us try to see how this expression actually depends, or what is its dependence on the partial pressure of the CO. So, suppose if I plot PCO versus CCOS, then the typical curve is that you have initially it actually increases linearly and then it actually remains constant. So, this region is actually linear, and as the partial pressure of CO increases further, the concentration of the adsorbed species is no more a function of the concentration or partial pressure of the gas CO. So, initial phase where low concentration it behaves linear, and that sort of can be seen from this expression. One can actually deduce from this expression that if PCO is very small, then Ka times PCO is much smaller than 1, which essentially means that it behaves like a linear function with respect to PCO. So, now how do we know what is the adsorption isotherm? The best way to do that is to postulate an isotherm, postulate a model, for CCOS versus the partial pressure of CO, and then validate with experimental data. So, the postulation of a model is what we just did a few minutes ago, and so suppose let us say I have such a model where CCOS is essentially given by Ka into PCO into Ct divided by 1 plus Ka into PCO. Suppose I have this model, then I can actually rewrite this model, rewrite this expression in the form of rewrite this expression by essentially taking 1 by CCOS, you take inverse of this divided by CO into CT, and this I can actually rewrite as, so this expression can be simply rewritten as P times partial pressure of CO divided by CCOS is equal to 1 by Ka Ct plus PCO by Ct. So, now if the experiments are done at different partial pressures of CO, so I can now plot CO versus PCO divided by CCOS, and based on this expression I should actually get a straight line fit. I should get a straight line fit whose slope is equal to 1 by Ct and the intercept should be equal to 1 by Ka into Ct. So, if the model that I propose is essentially the molecular non-dissociative form, then I can actually use that model and validate it with experiments using such a linear fit. So, let us now next look at the dissociative model. Let us look at the second case of dissociative adsorption. Let us look at dissociated adsorption. So, we can represent the adsorption of CO in a dissociated form as a reaction, as a reversible reaction step, where we have CO plus 2S reversibly gives CS plus OS. So, now I can write the rate of attachment is essentially given by Ka into partial pressure of CO into Cv square, and the rate of detachment will be given by K minus A into concentration of Cs into concentration of Os. And the conservation rule now is Ct will be equal to total concentration of sites will be Cv plus the concentration of sites occupied by C plus the concentration of sites occupied by O. Now, since the active sites containing C and O essentially comes from this species, for this case C of Cs should actually be equal to C of Os, and so from here we can see that the conservation rule is essentially equal to Cv plus 2 times Cos, and the rate of detachment is K minus A into Cos square. Okay. So, now let us plug these into the rate. I can now write the rate of adsorption as Ka into PCO into Cv square minus K minus A into Cos square. So, from here I can actually rewrite this expression as Ka into PCO into Cv square minus Cos square divided by the adsorption equilibrium constant. So, at equilibrium Cos is essentially given by Ka into PCO to the power of half into Cv. So, if I now plug this into the conservation rule, I will see that Ct is equal to Cv plus 2 times Ka PCO to the power of half into Cv, or from here Cv is equal to Ct divided by 1 plus 2 Ka PCO to the power of half. So, this I can now substitute in this expression here. So, if I substitute in this expression, what I find is that Cos is essentially equal to Ka into PCO to the power of half into Ct divided by 1 plus Ka 2 times Ka PCO to the power of half. So, once again I can actually modify this equation to bring it into a linear fitting form, and the way to do that is I once again take an inverse, and if I take an inverse I will see that it will reduce to the form where it will be PCO to the power of half divided by COS is equal to 1 by Ka to the power of half into Ct plus 2 times PCO to the power of half divided by Ct. So, I will get this linear relationship expression between the partial pressure of CO and the concentration of the oxygen molecule which is attached to the sides. So, now I can now draw a linear fit. Suppose if I have experimental data where I actually measure the concentration of the oxygen attached to the sites as a function of the partial pressure of CO, then I will be able to draw a fit where I can actually draw PCO on the axis here to the power of half and PCO to the power of half by CO dot S, and I should actually get a straight line fit with the slope being 2 by Ct and the intercept being 1 by Ka to the power of half into Ct. So, this is how one can actually measure the experiment, use the experimental data and validate the postulated model, and if the experimental data matches well with the model, then we know that the postulation is correct, and then we can actually verify the postulate and we can say that that is the isotherm. Okay. So, such kind of an isotherm is what is called as Langmuir isotherm. So, what we have just derived for two cases of dissociated adsorption versus non-dissociated absorption is essentially we have derived the Langmuir isotherm, which essentially quantifies the amount of gas that is actually adsorbed onto the active sites at different pressures and at a fixed temperature. It gives you what is the relationship between the concentration of the adsorbed species and the partial pressure of the gas. That is what is called as a Langmuir isotherm, and such an isotherm is actually very useful in order to understand how the adsorption process happen and how to quantify the adsorption process. So, next let us look at the fourth step, which is essentially the surface reaction term. And note that in the previous case where we derived the Langmuir isotherm, what we have actually implicitly assumed is that the active sites are uniformly distributed. So, we have implicitly assumed during the derivation that the active sites are actually uniformly distributed. So, let us now move on to the fourth step in the catalytic reaction process, where it is a surface reaction. So, now the question is, if what happens, how do you find the rate at which the surface reaction happens, and if I understand the rate, if I am able to relate the rate to the concentration of the adsorbed species or concentration of the measurables, then I have found a way by which I can actually quantify the surface reaction process. So, let us look at how to do that. So, essentially there are many ways to quantify surface reaction. So, we will look at how to find the rate at which the surface reaction happens for a few cases. So, we will specifically look at single site case, we will look at dual site case, and then we will also look at what is called as the Elie-Riedel case. And even in the dual site case, there are actually three different cases we will look at, where you have one adsorbed species, you have multiple adsorbed species, and then we have dual catalyst case. We will look at each of these cases where we will try to find out what is the rate at which the reaction happens. The surface reaction happens for a single site case, we will look at it for a dual site case, and we look at it for a Elie-Riedel case, where in dual site case we look at 3 different possibilities. So, what we have essentially covered in this lecture is we attempted to quantify the adsorption process. We attempted to find out what is the rate at which the adsorption process happens and how to relate the concentration of the species which is adsorbed onto the active sites versus the partial pressure of the species in the gas phase. So, in the next class we will start looking at how to quantify the surface reaction. Thank you.